Okay, and then Dom, whenever you're ready, you can start screen sharing. So the talk should last around 45 minutes, and then there will be an opportunity for questions at the end of the session.
Okay, hello everyone. Thank you so much for having me here. It's really a treat and an honor to be part of the Centre Seminar series. This is actually, I think, my first talk I've given since I've joined UCL, so I'm really excited that it's going to be here. And please be nice to me. I'm out of practice. So today I'm going to talk to you about this concept of of open neuroscience and working with secondary data. And the thing that I'm really excited to talk about is this idea of a multiverse analysis. And if you're not sure what that is, I'll get to that. But there's a little bit of a story before we get there. Before I talk about the main topic of today, I want to give a little plug to my other line of research. There you go. So my research is broad. This is a kind of my neuroscience is kind of my side hustle. My main thing that I'm really interested in doing is using open science and advanced quantitative methods, whatever they may be, to look at how our daily daily manifestations of our cognition, what affects them, what shapes them, how daily experiences affect things. Here's just a couple of things from papers I published. On the left is this model where we combine lots of cognitive measurements that we've measured, the cognitive variables that we've measured every day for 75 days, along with some non-cognitive things. And you can see how they're related. They're all pointing to each other. Different lines mean whether it's positively related, whether it's negatively related. On the right, you can see a measurement of mental rotations, of 3D mental rotations, we recorded every single day in participants for 75 days. This is called a spaghetti plot. This is an amazing graph where literally everyone's data from the study is on one graph, and you can see lots of fluctuations. And my main area of research is trying to understand those fluctuations. I'm particularly interested in that in a classroom setting, an educational setting. I won't be talking about that today. But if you are interested in that, I'll be giving a talk at the start of July, which is more about my dissertation research and things like that. But for now, I want to talk to you about some research that I did during my PhD. These are just my collaborators. I thought I would give them a shout out too. I've had the privilege of working with some amazing neuroscientists, despite not being a neuroscientist myself. And all the papers that I've talked about today come from a group of graduate students that we formed during my PhD. And we were led by Dr. Keating here, who is a great guidance and advisor, but he mostly left us alone to make mistakes, to learn things, to do things independently, learn how you cooperate with people, learn how you collaborate. And it was a great experience. We were led by Michael Demidenko there on the left. I just want to say thank you to all of them. And the things that I'm talking about today, I hope I can encourage graduate students listening today that you can do this. You can get secondary data sets. You can get open data sets. And you can make collaborations with your colleagues and learn from each other. It's great to have mentorship as well, like we had with Dan. So yeah, you can do this too. So please do. Okay, so the things I'm going to be talking about today, I've split the talk into two parts. The first part may seem really simple at first, and it's a pretty basic question of what even is a replication? And a lot of you sitting here today will probably go, well, I know what a replication is, but I think I'm going to try and hopefully prove it's maybe a little more subjective than you might think. And then I'm going to give an example of a paper that I published that looked at these idea of replications in the context of this multiverse analysis. And the data set we used is the ABCD data set you might be familiar with. I'll talk a little bit about that. So in this first part, I'm going to talk about a lot of open science things. I, through one way or another, kind of become an open science person. But some of this might be assumed knowledge to some of you, but I think it's important to go over it and also to make linkages between certain concepts that maybe might be fresh. Okay, so it's not news to anyone that there is an ongoing replication crisis. It's been going on for quite a long time. I'd like to highlight here that it's psychology that traditionally gets most of the flack for this, and rightfully so in many ways, but it's not limited to psychology. This is an issue in medicine, it's an issue in pharmacy, it's definitely an issue in economics. It really is a multidisciplinary crisis, but I am going to focus mostly on psychology and neuroscience here today. So when can we say that something is replicated? Like I said, it kind of gets overlooked that people don't often agree on what a replication is. So some argue it's that when maybe the direction and the size of the effect is replicated, but does it need to be in the same direction? What about significance? Do you need to be under p is less than 0.05? I thought we were trying to get away from significance testing. Is that part of it? Are some replications better than others? If it's in a bigger sample and it doesn't quite replicate, is that better than a almost perfect replication in a very small sample? How do we judge that? It seems obvious maybe, but it's actually quite contested. Something I want to differentiate here is between what I consider reproducibility and replicability here. So for me, a reproduction is recreating a finding with the exact same sample and the exact same methods, while a replication, I would kind of consider it more to be determining the generalizability of a finding by repeating it in a new sample. A conceptual replication is when it's conceptually similar, not exact. I personally think what most people call replications are really conceptual replications. You might not agree with these definitions, and that's fine, because it turns out not a lot of people agree on what a replication is to begin with. I just want to give a shout out to this very timely report about reproducibility and research integrity. It literally came out last Wednesday, and in the report, which is a great read, it's not too long, I do recommend this, they also point out very strongly multiple times that among a huge experts of incredible opinions, that people don't really agree on what replicability is. That's partly because it's subject-based. Certain fields have different ideas of what replicability is, and the topic at hand. I personally quite like that definition, which is replicability research has been sufficiently open to allow for robust assessments of their work's data, methodology, and conclusions. And through such an assessment, is their research proven to be thorough and accurate? An emphasis on open there. This is a little bit of a tangent, but I'll go through quickly what some of their insights are. They point out that replicability problems are not necessarily bad practice a lot of the time, certainly not limited to social sciences. A lot of early career researchers felt that they had often been pressured to present a particular result for their professor or the principal investigator. They advocated the need for more slow science, which I definitely agree with, and a need for ref reform. It's a great report. I do recommend it. And I'll talk more at the end if you want me to talk about this. But again, I just want to emphasize that, again, it's not totally clear what replicability is, even among the experts. How often are replications attempted? It might not surprise you that it's not that often. So this is a paper here that used the entire publication history of 100 psychology journals. It's a little old now. I'm sure there'll be more replications in the last 10 years. They found that 1.6% of all articles used the word replication. About 2/3 of those were actual replication studies. Replications they found were more successful when the initial authors were involved. But it still comes back to a key disagreement in the field that they talk about in this paper, is that when is something replicated? Yes, numbers have definitely gone up in the number of replications since 2012, but even a slight uptick when you're starting at 1% is a drop in the ocean. So they're not that common. I also want to talk about this idea of what a verification report is. So verification reports started from, to my understanding, from a 2018 tweet. This guy, Sanjay, he's a great follow on Twitter, by the way. I highly recommend it. He talks about some great things. And his tweet said, in the same spirit as the Pottery Barn rule, for people who aren't as familiar with America, Pottery Barn rule is you break it, you buy it, right? The idea you break it, you buy it. We should have verification reports. There should be one-page online reports linked to the original, where you can report you ran the analysis to verify the numbers in the paper, and maybe do other analysis to verify the strength of conclusions. And it's this idea that if you're a journal, if you publish something, you're not just responsible for publishing that particular paper, you are responsible for whether it is replicable. You're responsible for how transparent it is, how open it is, and whether other people can create the same findings. So once you publish a study, journal should own its replicability, or at least a good part of it. So from that tweet, verification reports were born. Chris Chambers is a fantastic professor in Cardiff, and he came up with this idea of this verification report. It started, as my understanding, in Cortex, at least in our field, and it's where you repeat the analyses of a publication with the same data, or you apply new analyses to the same data. There are kind of rules to this. The 4 rules that are proposed by Dr. Chambers is that no members of the original team should be involved. This idea that by having a completely new set of people, maybe it's less biased, maybe there's less external forces trying to find a certain result. They should be reviewed in the same ways as registered reports. So for people who aren't familiar with registered reports, in registered reports, you first submit your literature review and your methods to the peer review. They get approved and your analysis plan. And then the second half is you do your results and your discussion. And then it gets, but in theory, if the first half has been accepted, the second half will be accepted no matter what the results are. And they're a great part of open science, and verification reports are organized in the same way. Not necessarily having access to the original data shouldn't be a deal breaker. There's a recent paper that came out, I should have cited here, that contacted people that say our data is available upon request, and found very, very few people actually make their data available upon request. So not having exactly the same data as the initial data shouldn't be a reason that you aren't allowed to verify things. You should be able to verify things by different means, by similar data sets. And the original authors should be invited to review it. It shouldn't be a completely external process to the original authors. They should be involved in some way. Ultimately, they did the initial analyses. There might be assumptions that people make that they can clarify with. So that's a verification report. There's not too many of them. I've highlighted two journals on the left that either do verification reports in neuroscience or something that's very close to a verification report. There are definitely more, but it's not very widespread thing yet. So this all works pretty fine for things, I think, for testing reproductions or things that are very close to reproductions. But when you start getting a little further away from reproductions into replications and conceptual replications, it might not be the perfect fit. So I just want to talk about what you might do instead. Okay, so the main thing I wanted to say here about verification reports, about replicability, about open science, and if you only take one thing away from this talk, I really want you to take away this statement, which is data are not passively collected. We construct data in the vast majority of the time. Observational data may be slightly different, but even then, there's a level of construction involved. And by construction, I mean, we as researchers, we have to operationalize things. We don't have data just handed to us in a particular format, and we know this is the purest, most perfect form of the data. We make decisions, consciously or unconsciously, about the data that we collect. It may be as simple so you don't even think about it, whether you have a data expressed as continuous or categorical, maybe make it a median split, maybe you have an exclusion criteria of, a Z score above a certain number instead of another number. These are all decisions. And that's not even to get into if you have a general measure like stress, which instrument you use, which measure you use. These are all decisions. We construct data. And that's important when we think about replicability, is an acknowledgement that we are all constructing data in our research, whether we want to or think about it or not. So this leads us to the idea of the garden of forking paths, which is the idea that as researchers, we often explore the many paths and opportunities available to us, even if we don't acknowledge it. So we can make many decisions that affect our outcomes of analyses. These are often called researcher degrees of freedom, right? It's like degrees of freedom, but in terms of a researcher, that for any given decision, any given measure, any given statistical analysis, we often pick between different things in an exploratory framework, or maybe some people would call it some other things. Often people do try out different combinations of variables or try out different statistical analyses, but they only end up reporting the ones that they want to report, whether it because they're significant or whether it matches the original hypotheses. That's often called p-hacking or phishing or things like that. But even if you're someone who doesn't do that, even if you do think completely by the book, there are still ways, there's still potential comparisons that we don't even make, that we don't think about. The idea of the forking paths is, even if we don't try all the different routes, they're still there available to us. There's still possibilities that we could have explored that we didn't. And all these different roads and all these different tangents, different forks, lead to many, many, many different ways that a study could have been done. So we have to acknowledge there are potential other studies that don't exist that we also have to compare to. And there's just many ways we can handle variables. And then you have this other question of, if you publish a solution and maybe you only looked at one different path, maybe you didn't try lots of different things, you kind of did what you said you were going to do, maybe you pre-registered it, maybe you followed that line of research. What happens if what you published, which maybe was significant or however you want to measure success, what if that was the only solution? What if that was the only combination of variables that gave you the conclusion that you made? What if you had done that one tiny thing differently and you didn't get the same results? Sometimes we're aware of these things. For example, a very common one is if there's a clear outlier, we might test our analyses with or without the outlier to make sure it's not affecting things like that. But often there is I don't want to say infinite, but a very large number of things we're not even aware of. And no other open science thing will help you here. You can say, well, I pre-registered, I registered reports, and that's great. But that means you did only explore one route. What about the other routes? Would you have got the same thing? I don't know about you guys. I publish that much, but when I do, I sometimes kind of lie awake at night and thinking, what if I did something differently and I would not have got the same results? And there are theoretical reasons you might justify one path over the other, right? You might, hopefully you sit back and go, well, it was the right decision to go down this route because of X, the right decision to operationalize something this way because of Y. And that's great, but sometimes there are decisions we make that could have been argued either way. And if it only works in one combination, what does that mean? Is that a problem? Now I want to take a stop here that, again, this is an open science talk, and you might say, Dominic, I kind of paid attention to an open science talk before when I wasn't on my phone. And I remember everyone saying, you do not do different combinations of things. You do one thing, you pre-register it, you follow that path, you do not explore things, you do one thing. You do not run every combination possible. That is a bad thing. That's exploratory, it's p-hacking, blah, blah, blah. I don't disagree, but I want to revisit that point in a few minutes. Let's think about that. Is it the right thing to do to explore different combinations of variables? Should we just stick with one and never look at one of the other opportunities? We'll think about that later. Okay, so we've established that we really don't all agree on what a replication is. Maybe it's like the famous, I think it's a Supreme Court case in America about obscenity, where the person said, I don't know what it is, but I know when I see it. Maybe that's what replication is. Maybe there isn't an objective way of determining it. But let's take things a little further. I would like to now talk to you about an example that I've published that's to do with replications and the difficulty that we had when determining when something is replicated. And it's a pretty extreme example that involves many, 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 many different potential replications. Okay, so the data set that we worked with is a data set that many of you are, I'm sure, are familiar with. It's the Adolescent Brain Cognitive Development data set. It's run by the NIH, the National Institutes of Health in the US. And it's a great data set. It launched about 5 years ago now. It's a 10-year cohort study of about 12,000 children from across the United States. It's a really rich data set. It's a combination of imaging, and it also has a survey component. There's lots of different types of imaging involved. There's structural imaging, there's functional imaging. Children complete tasks in an fMRI machine. There's also some resting state scans, lots and lots and lots of different surveys. It's really rich. The baseline started in 2017 when children were 9 to 10 years old, and the intention is it'll follow them up into early adulthood at about 19, 20 years old. We're up to release 5, which time flies. The paper I'm going to talk today is about releases 1 through 3. And it's fairly representative as it goes. In terms of American data sets, often they're only from the Northeast, so they're only from the Bay Area. This is across the whole country. It's approximately representative in terms of gender, in terms of race and ethnicity or socioeconomic status. It's not perfect, but it's a lot better than other studies of its type. When the data was initially released, the first baseline measurements when children were 9 to 10 years old, I just want to mention that not all the baseline data was released at the same time. There was an early release, and just follow that in the back of your mind, because it's open data set, people can really start working on it whenever data is available. And there were some people that did analyses with the first release, which is only about 30% of the data, while some other analyses waited until 100% of the baseline data was out. It comes to an interesting point, I suppose, of in academia, you're rewarded for doing things first or maybe doing things best. But even then, I think first is the one. And so when you had so many people working on the exact same data set at times, and there's a limited number of topics in the world, maybe at times it felt like a bit of an arms race to be the first to get to look at a certain set of data. And I completely understand why people did. But it introduces an interesting question when it comes to replication, that even in this huge data set that in theory is available to everyone in the exact same way, people weren't necessarily always working off the same samples, which I think is interesting. Okay. Yeah, so a general fact that I'm sure most people will here will agree with, we know that environmental factors meaningfully impact the brain. The research group I talked about at the start, of amazing people, that was the main topic that really interests us. We came from very broad backgrounds and very broad interest areas, but this idea, we know the environment affects the brain. We know adverse family environments have been reported to affect brain maturation and pubertal onsets, for example. Early pubertal onset is related to earlier brain maturation. We also know this might depend on specific experiences. It might depend on child sensitivity to environmental context. But beyond this, we know that there are also significant debates about how we should characterize these environmental stresses. Again, it may seem pretty pretty obvious how you characterize it to you, but it's done in a wide range of ways. Some people use individual scales to characterize the environment. For example, family to needs ratios, their poverty level, parental education, area deprivation indices, and they might use one of these, two of these, three of these, a couple of variations of these. While other people might use latent factors, which are composed of lots of different scales, maybe put in some kind of confirmatory factor analysis framework, and it might affect a broader construct. For example, general levels of neighborhood stress, general levels of deprivation. Which one of these is better? I'm sure a lot of people watching this now might have an opinion on what one is clearly better, but there isn't necessarily a lot of agreement in the literature. And again, returning to this idea of replication, this might affect our ability to successfully replicate things when people do things in such different ways. Okay, so as a research group, the first paper we produced on this topic was published in Cortex. It used the ABCD data. You can go read it now. And we use the share interest in environmental factors in the brain. I'm really proud of this paper. It's the first paper in neuroscience that I published, the first paper we came out with as graduate students. You could argue it's a little bit overambitious, and I think that's probably true, but I think it's a great paper with lots of things in it. It was a great introduction to the ABCD data set. It was my first time working with a big data set like that. And although this talk is about a different paper, I wanted to give you a little bit of a, as they say, TLDR, a shortened version of what the paper is about. So what this paper is about is the idea that children experience more ecological stress growing up. Few studies have found that they also have symptoms of internalizing problems. They also have more a reactive amygdala. A few studies have found that, but they're usually in small samples because neuroscience samples are usually pretty small. It's really hard to get people into a scanner. So we thought now we have the opportunity to test it in a huge data set like the ABCD of whatever it's 11,000 people. We also have the opportunity to test some confounders that maybe weren't necessarily tested in small samples, or maybe wasn't well powered enough to look at. So we took the ABCD data, we looked at the 9 to 10-year-olds, which was the baseline, and we also looked at the first follow-up, which was all that there was released at the time. We measured amygdala activation with an emotional end-back task, specifically the faces versus places task. And what we found is a significant association between ecological stress and internalizing symptoms. We found no association between amygdala activity during the faces versus places contrast and ecological stress or subsequent internalizing symptoms, and no mediating role of the amygdala or moderating effect of parental acceptance on the association between ecological stress and internalizing symptoms. The paper is way better at describing that than I did, but I hope that's a shortened version. But based on this experience of doing this together, what we actually found is that we weren't alone in doing a study that sounded a lot like that. So there were actually quite a few preprints that use the exact same ABC data or pre-registration, so you can look on OSF, that looked at pretty similar questions. And that makes sense because there were hundreds, thousands of researchers all accessing the same data, often with similar questions, purely by coincidence, which initially was a little disappointing to us because you always want to be first, right? You want to be novel, and finding out other papers were kind of doing quite similar stuff to us was disappointing. But the more we thought about it, we kind of realized, well, they're not doing the same thing because even though we're looking at the same latent variables, the same latent constructs, they were measuring things in actually very different ways. In a huge data set like this, the ABCD, for example, something that is very popular construct like socioeconomic status, they measured it in many different ways, right? That's not uncommon. You see the same thing in Mediating cohort study and things like that. Again, internalizing problems in the ABCD data was measured in many different ways. So you have all these researchers looking at pretty similar questions, but combining their variables in very different ways. In the ABCD, you have income, you have area deprivation index, you have neighborhood safety, just a few things. We, for example, in our study, did a confirmatory factor analysis to make a latent variable of environmental stress, but others didn't, and that's a perfectly valid decision. Even within one variable like income, it was presented in deciles, you could have median split that, you could look only at the top 20% and the bottom 20%. So even multiple studies studying income were doing things in a very different way. And sometimes not even with the same sample. You've got this issue, like I mentioned, of the early release and the full release, but also different studies have different exclusion criteria. You might exclude certain participants due to excessive motion, but other people might not be as stringent about it. Even the samples weren't all the same. They were pretty similar, but they were different enough that you could reasonably expect different conclusions and different analyses. So this process of working with the ABCD data in this first paper, it kind of left us more questions and answers about the process of working with a huge open data set like this. And I would love to sit back now and tell you, well, I'm confident that we operationalize everything the right way and everyone else did it the wrong way, but that is obviously not the case. There are reasonable reasons you would make certain decisions, and there's strong theoretical reasons to do things. So it left us in kind of thinking a lot about, does it matter how we operationalize things? Okay. So this moves me to a wonderful thing, which is the multiverse. If you have seen any of these 3 movies on your screen in the last few years, you know about this concept of the multiverse. For the uninitiated, is this idea that you have characters from different universes that look a lot like the characters you know, look a lot like the Spider-Man you know, and they cross universes, cross paths, and come together. And they're recognizable to us, but a little dissimilar as well. In reality, a lot of us also, since about 2016, might have thought we've also entered some kind of multiverse different timeline. I'm not sure we have, but it's definitely a phrase that has passed into our lexicon. But the general concept is the idea that there are universes that are not dissimilar to ours, where they're pretty similar, but there's some slight changes, and they might have big effects. So let me tell you a little bit about the idea of a multiverse analysis, kind of building on this. So it was proposed initially in a paper from about 2016, and it's this idea that papers don't have to consist of just one analysis. And we know that single data decisions are often arbitrary, or they lack justification, and actually single analysis can be misleading. And so a multiverse analysis performs not just one, but all of the reasonable scenarios for this analysis. So this is the paper, by the way, you should read it, it's a great paper. So a multiverse analysis gives you an idea of how much your conclusions might change due to your researcher degrees of freedom, due to operationalizing things in a slightly different way, or making a latent factor, not making a latent factor. And it'll tell you a bunch of things. But one of the things it'll tell you is, in some ways, how fragile your conclusions are. And again, if you're worried this idea of being kept up at night of not having done things the right way, getting a better idea of that fragility of your conclusions, how much you can really hang your hat on them, is really important. If you can break them pretty easily by doing justifiable things, maybe you won't be so sure about the conclusions. When we talk about generalizability, we usually talk about it across samples, right? And that's obviously really important. But it's also important for conclusions to generalize across operationalizations. And again, you may not agree with every operationalization. Some are just wrong for your sample or wrong for your study. There's no reason to test those. But anything that is reasonable, and that word reasonable comes up a lot in this paper, might be worth testing. And you can end up with a lot of different combinations. If you just take a theoretical example, let's say your independent variable, there are maybe 5 ways you could have operationalized that. A dependent variable, maybe 3, maybe mediator, 4. Maybe you could have had 2 different covariates. 5 times 3 times 4 times 2, hold on, I can do maths. 120 different combinations. So there's loads of different universes that these analyses have been conducted with, and you might be interested to know, well, how well does this generalize across all these different universes? Or the phrase they use actually in the paper is many worlds, which is a good one too. I just want to talk a little bit about this. So you might reasonably say to me, Dominic, if I do 120 sets of analysis, how am I meant to communicate this? And no one is going to read 120 sets of results. I'm not going to read 120 regression tables. What am I going to do? So the way that these are often displayed is on something called a specification curve. It's a great way of communicating this. I've given the credit to down the bottom there to someone who gives a great tutorial online about how to use these. So I'll walk through what this is. So a specification curve visualizes the strength of the effect between your constructs or constructs of interest across lots of different models and the analytic decisions that you made. So it's split into 2 parts. The top graph here, it may look like a solid line, but it's actually not. It's actually lots and lots and lots and lots of little dots. And each of these dots is a different regression coefficients based on a different multiverse analysis. They are ordered. So you can see they're ordered in terms of standardized regression coefficient here. So they make something that looks a lot like a line. And it gives you a great idea of the amount of uncertainty. If you have something that was not very uncertain across your, whatever, I think it's like 400 here, you would have a pretty flat line, or it wouldn't change that much. In a case where you have kind of like here, you have a lot of differences, that implies there's quite a lot of uncertainty. Remember, each of these is a different combination of variables or a combination of decisions. Down below, these are the different decisions. I've cut off a little bit, it's quite long. But you can see that each of these little lines, that is a different combination. It's a different combination of variables, and you can color code them or sort them as you have way you want. So this is just a way of showing that of what results you get in a kind of clear and I like to think communicative way. Okay, we're going to stop again. Okay, but you told me again, you're not supposed to do every analysis. That's a fundamental thing of open science. This can't be right. Did I see Andrew Gelman's name on that last paper? Is that a different guy? How could he approve this? So I guess for better or for worse, I am a little bit of an open science guy now, and I get a lot of questions about how to do open science the right way. But my stock answer, which is the answer which is in that paper I cited earlier, is it's all about transparency. Everything here is about transparency and being open. The problem isn't necessarily running lots of analyses. The problem is running lots of analyses, but covering them up and pretending that you didn't do them. And in a frequentist statistic framework, making generalizations based on the fact that you ran many, many analyses when you pretend that you did not. It's about transparency. It's all just about being open. It's fine, as this multiverse analysis shows, it's fine to run things in every possible different way, as long as you're transparent about the fact that you do it, as long as you're transparent about the fact that you're not selectively reporting, and you're emphasizing to the reader that the reason that you did it is to improve your science, is to be more confident in what you're doing, and to emphasize at times the arbitrary nature of lots of decisions that you made. So in terms of something that I'm particularly interested in as an adolescent research, which does have a bit of a measurement issue, is puberty. Puberty might seem like a pretty standard thing and ways of measuring it, and in some ways it is. The gold standard for measuring pubertal development has been for the longest time is the Tanner stage, which is that for people who don't know, you compare a child's pubertal development against a certain scale. There's different ones for boys and ones for girls. The gold standard is administered by a medical professional. But it's also considered quite intrusive. It's also very time intensive. I don't know this, but I'm guessing it's not included in the ABCD data set. I'm going to guess the reason is for those exact reasons that it may not be ideal for retention, and it takes a lot of time, and people didn't necessarily have a lot of time with all the participants. So in the ABCD data set, it is self-reported. But here's a catch. It's actually self-reported in two ways. It's self-reported by the child themselves. They are asked questions and to compare themselves to the Tanner stages. It's a well-validated scale. And it's also reported by their parents. So we have two different ways of doing that. And again, as you might expect, puberty is a pretty popular topic. And in the papers that are out there, people use different versions of this. And some papers actually use a hybrid where they'll average the parent reports and the child reports. Here's another thing that we found in our paper. They're actually not well correlated. The correlation is about I believe is about a correlation of 0.2. You might go, it seems strange to me. You're talking about the same thing. How can it be so low? Well, why might they disagree? I'm happy to discuss that more at the end, but there are different reasons you might pick one over the other. In terms of the adult sample, you might think, well, they know best. Children don't really know what pubertal development is because they haven't gone through it. Well, adults know what it looks like. They will be the better judge. But on the other hand, you might say, well, it's always better to ask people themselves about measurements. They will know, children will know themselves better than anyone else. So maybe you should trust that measurement. So if you have this two measures that don't correlate that well, you have this issue of which one do you go with? And this is the thought process that really led us to this multiverse analysis. Okay, so knew we wanted to do a multiverse study. We knew it wanted to be one that used puberty as a measure. We knew we wanted to use the ABCD data because of its rich brain data, and there were many to pick from. And if we were trying to find one we didn't like very much or we didn't think was very good, it would kind of be like shooting fish in a barrel. There are quite a few we didn't like. But we thought, why don't we actually pick a study that we respect and that we thought was well done and to see if it replicated? In some ways, what's the point of replicating a study that we didn't really believe in the first place? So we wanted a study that we believed in. And so we picked this. It's a great paper written by Sandra Thiessen and Paul Collins and Monica Luciana. These are great researchers who put together a paper that was one of the first to come out the ABCD data and one of the first about puberty. And it's a great read. I recommend it highly. So this is the study that we decided we want to try and replicate in some way. So I'll just walk through it. So the question they asked is whether the family environment affected amygdala medial prefrontal cortex activation, and was it mediated, crucially, by pubertal development? Their method was the early wave, so not the full wave of the ABCD, the early wave of about 3,000 pre-adolescents. They found indirect effects, which suggests that more stressful households led to more advanced pubertal structure that itself led to more mature brain structure. They found that this result was stronger in girls than boys. So it's a good paper, and these are solid findings that I believe other people have either speculated or found in smaller samples. But there are decisions that were made during this paper, as there are in every single paper. So they used, as we mentioned, only 38% of the full sample was used here. That's all that was available at the time. There were quite a few propositions of research degrees of freedom. For example, which measure of pubertal development did they use? They used the parent one for the record, but they could use the child one. All sorts of, in the brain was the dependent variable. Which element do you use? They do full functional scans, they do resting state data, structural elements. There are lots of things that they could have picked. And when they were measuring the family environment, which is your independent variable, they made a latent factor, very similar in the paper that we did, of looking at parental conflicts, monitoring acceptance, parent reports of conflict, and psychopathological variables. I think this is a very justifiable approach. This is kind of comes from a cumulative risk approach, which is one I myself agree with. And I think it makes sense, but it's not how everyone would do it. Some people on similar topics have used just one variable or just maybe a combination of variables. You can see where I'm going at here. There were things that we could explore through a multiverse analysis. So we did. You can read this paper in Neuro Image Reports. I will walk through the two parts of it. It's a two-part study. The first part is a replication of the original study, but with the full baseline data. So operationalizing things the same way they did, but in the full sample. And then the second part is a multiverse analysis to investigate how different ways of operationalizing family environments and puberty may impact conclusions about the brain behavior associations. You can see that the two of the authors, Sandra and Monica, are on this paper. The eagle ears of you may notice that I said earlier that one of Chris Chambers' tenets of verification reports is that the original authors should not be involved. And you might also remember from way back at the start of this paper, that I said replications are more likely to be involved if a member of the original study is involved in replication. So in that sense, this isn't a true verification report. This is what is called instead an open data replication, Neuro Image Reports. Why we consider having the original authors there to be a great thing. We consider it to be a massive benefit. They were a great help to us. Is there sometimes bias having the original authors? Maybe, but it depends who it is. Monica and Sandra were incredibly respectful. They very much sat back. They wanted to know what we found. They wanted to know themselves whether their research replicated. They wanted insight into what we could do. And beyond that, we wanted to include them because we didn't want the replication process to feel like accusatory science. We wanted it to feel like a collaboration. I think of a lot of people watching this right now who heard that someone was out there trying to replicate their most cited paper. Your first feeling might not be great. Your first feeling might be fear or a sense of someone's out to get you. I personally don't think that's healthy. I think making this a collaborative process is a good thing. And so we were very happy to have them involved. And we can talk about at the end. I think that's the way forward for the slide research. Okay, so I'll walk through the first aim. So again, the first aim is replication. So in the full sample, instead of the smaller sample, it's a pretty simple mediation model. You have the family environment. The mediator is parental reported puberty. And then there are 5 different brain-dependent variables, which you can read on the right there. The family environment factor, remember, was a latent factor. So in other words, the core of this analysis was to test the indirect effect of reported pubertal development on the association between the environment and between structure and brain structure and function. What did we find? Okay, but before we talk about what we find, I want to say that we were left with a question of how do we decide something is replicated? Fundamental to this aim is making that decision. And you might say, well, it's a binary outcome, right? Someone asks you, did this paper replicate? The answer is yes or no, that is it. But it's more complicated than that. So we followed Open Science, OSF, Open Science Club guidelines on this. And we evaluated them based on 3 criteria. The first criteria is, are the direct and indirect effects consistent? We looked at the direction of the effect. We looked at the significance of the effect. Are they consistent? The second one is, how do the effects of the replicated study overlap with the confidence intervals of the original and vice versa? So we looked at that. And then the third one, by far the most subjective one, is, do the authors subjectively think it replicated? So the people who are on the study, who are hopefully experts in the field or close to it, in their opinions, is it a replication? That was decided independently. We literally just took the data we had, we looked at the results, we went away separately, and we wrote ourselves for every direct and indirect effect, did it replicate? I want to talk a little bit about that last one in particular. So what's interesting about this is we didn't give guidelines, really. We just said, here's a spreadsheet, Just write the words yes or no into every cell for every decision. That's it. But despite not having that framework, we came together afterwards and we talked about what we did, and we found out that we actually all used 2 evaluative techniques. I believe 5 of us did this process. So the first one was you either interpreted whether the effects and the magnitude of the effects between the original and replication study are the same. That was the criteria. And then the second group of us kind of, as well as that, considered whether changes in p-values might impact the takeaway by the readers or the publishers. It wasn't based on was it significant or not. It's just, is the change in significance enough that it would affect your understanding? Is that the right way of doing it? Happy to talk about that later. So what did we find? So the first way, again, is we looked at whether the effects are in the same direction and same magnitude. So this graph shows on the top, it's the confidence interval of the original study with the replication placed next to it. The replications are in X and the original study is represented by an O. And then below is the other way around. We have the confidence interval of the replicated study, and the O is the effect of the original study. So across the 5 brain regions that we measured in both the original and the replication study, 90% of the estimates were in the same direction, and 60% of the p-values were in the same significance category. So we saw direct effects of family environment on the brain and indirect effects of pubertal stage linking family environments in the brain that were significant in the replication study, actually weren't significant in the original study. So although 90% were the same, 90 and 60% were the same, there were some things that were significant in the replication that weren't significant in the original sample. That could be down to power or whatever it may be. The second way of looking at things, we looked at the confidence intervals. So what we found is that the beta estimates from the replication models overlapped 100% of the time with the 95% confidence intervals in the original paper. But only 50% of the beta estimates from the original paper overlapped with the 95% confidence intervals from the replicated models. That makes sense. It makes sense at the larger sample, at the smaller confidence intervals, I would say. So it's interesting, I think, that it's that way around. And this also shows the importance of testing both directions and not just using confidence intervals of one. The third one is the subjective one. So in terms of agreement between raters, the raters determined that 60% of the effects replicated in their subjective opinions, 10% did not replicate, and on 30%, we could not come to an agreement. We just could not decide. And that's a small group of 5 people. So imagine what it is in a huge group of people. So anyway, those are the 3 ways that we considered, did this study replicate? The second aim was about the multiverse. On the left, you can see the list of all the variables we explored. You can see there's 9 for environment. You can see that there's 3 for puberty and there's 5 for dependent variables. 9 times 3 times 5, quick maths, anyone? 135 possible models. And on the right, you can see the model that we explored is the same as aim 1. It's a mediation model. It's just instead of having 1 environmental independent variable, there are 9. Instead of 1 puberty factor, there are 3. And so we tested every possible combination of these 17 different variables. Okay, so these are the analyses. So this is a specification curve that we talked about earlier. And this on top is a specification curve for all 135 of the models. Blue circles are significant and a positive effect. Red are significant and a negative effect. Gray are non-significant. Looking at this curve, you can see that roughly 1/3 to 1/2 of the time, you see a mediation where there's an indirect effect of puberty between environmental factors and brain-dependent variables. These are all indirect effects. And to be precise, across all 135 models, well, actually, sorry, they're not all indirect effects. 39% of indirect effects were significantly different to 0, 42% of direct effects, and 45% of total effects. So those are pretty similar numbers. So across the board, it's about 2/5 of the time we found effects significantly different than 0, no matter what combination of variables you used. On the bottom, those are all the different combinations of variables. Then the second element of this is we looked at the standardized beta estimates for the direct and indirect effects. This is only actually 1/3 of the total number. This is the parental reported puberty models only. If we put all of them on there, it would look very messy. Remember, puberty was measured in 3 different ways. This is just one. So the family environment beta and its associated 95% confidence interval is reported in both panels. That's to provide a reference for how much the effects diverge from the original model. So we want to put the original model in there as something to compare it to, depending on how you operationalize all the different variables. Relative to each other, there was comparatively greater variability in magnitude among beta estimates for the indirect effects, particularly MIG-COM. Regarding the factor-derived scores in particular, there were significant results for all 4 predictors, for the ACC-CT brain region and for left and right and MIG-COM. Okay, and then the last bit of this is we wanted to get really into this puberty operationalization, which motivated all of this. So across all the direct effects, across all 135 models, as I mentioned, there's about, doesn't matter really whether puberty was parent-reported, whether it's child-reported, whether you average the two, you got about 40% of the time, maybe to 45% of the time, you saw a significant direct effect. In terms of the indirect effect, how you operationalize puberty really matters, because for the parent-reported models, 76% of the time you found a significant indirect effect. Sorry, I should say 0% of child-reported models did you find indirect effect, and 38% of the time when they're combined. That's a stark difference depending on whether you use parent or child to puberty as your mediator. Okay, so conclusions. Although we replicated most of the estimates in the original study, we have demonstrated here how different operationalizations can result in different conclusions about how puberty mediates environment and neurodevelopment. Different ways of understanding an adolescent's home environment during puberty can potentially result in very different practical suggestions for interventions. And we generally caution developmentalists and education specialists from interpreting which effects are meaningful using only a traditional null hypothesis testing framework. Only using significance here will give you a very different set of results than if you look at other things and the different ways we did things. Okay, so lastly, I'll end with these 2 takeaways from this entire talk. Number 1, I still don't know what a replication is. I still don't know. And in this paper, we explored it in multiple different ways, and all the results are basically, yeah, it mostly replicated all of these things, but a couple of things didn't. Could be explained by the larger sample, but not entirely that. If you were to ask me binary, did this paper replicate? I'd say yes, I would say that. But I don't think a binary way of describing it is necessarily the best way of doing it. Maybe that's okay. Maybe it's okay we don't all agree on what replication is, because maybe it's contextual. Maybe we shouldn't be thinking of replication in a purely binary sense. Maybe it's okay, it's subjective. Maybe it requires contextualization, and that's a good thing. Maybe these replications are not providing a clear answer. Maybe it's just adding to a picture, and that's okay too. In the original paper about multiverse, there's a quote I really like. I think it's the last line of the paper. It says something like, performing a multiverse analysis will often be difficult. It definitely was. And to a large extent, subjective. And it was subjective. You can look at those slides and think you would have done it differently, and you're not wrong at all. But it does not, maybe there should be researchers' degrees of freedom for multiverse analysis. But that doesn't change the fact that this is part of transparency. This is meant to be hard. We're meant to disagree with each other, but it is still useful towards transparency. And the other takeaway I want to give is this should not be a witch hunt. I think there are people who are scared of people trying to replicate their study, and I understand why. It feels like a lose-lose situation. We do have to change that climate. Maybe sometimes people do find errors somewhere, or maybe there are operationalizations that might be more generalizable if they'd be done differently. That's fine. We need an academic culture where that is accepted as fine, where no one thinks they're going to lose their job just because they put their data set out there as open data and someone else takes a look at it. Replication are a healthy part of our scientific climate. It's not something to be scared of. And I'd like to encourage you all, especially with open data sets, secondary data sets that are available right now, to do it yourself. I'd like to say thank you very much to all the people who took part in ABCD, my collaborators. If you'd like to talk more about it, please do contact me, and look forward to your questions.
Thank you, Dom. That was a really interesting talk. Lots of maths for this time in the afternoon. Lots of food for thought. So yeah, I'll open the audience up to questions now.
So should I put my speaker?
We've got a bit of an issue here because there's several of us in the same room with laptops, by the way, to the audience. I'll start with a question while we have a moment for other people to post their questions in the chat or raise their hands. So I was wondering, how do you then communicate your replication results to the general public? It's very complicated stuff, and you did a really good job of explaining it to us today, but it's sometimes not satisfying. You know, it's not going to make newspaper headlines having this kind of ambiguity. So
I agree, it's really hard. The thing that we thought about a lot when writing this paper is, what's the elevator pitch? Or, sorry, in Britain, the lift pitch, where you're at a conference, you're at SRCD or IMBUS, and someone goes, oh, I hear you tried to replicate that paper. Did it? And ultimately, it's really nice to say yes or no. And for the most part, I think you can say yes or no. But you're right, you can't show similar specification curve, and you certainly can't show a member public specification curve and go, well, as you can see, 60% of the time it replicated, but it didn't. It's something I think we need to work on, but I think it's okay to say to people, did it replicate? Mostly yes. I think that's an acceptable answer. And you can put caveats on that. You can say, mostly yes, and I would say it might be because we have more power or this or that. But yeah, it's a tricky one. But part of it is we have to be able to accept shades of gray, which is tough, and we as humans don't like doing, but it is part of it.
Great. And we've got a question from Zara.
I have two questions.
Okay.
So parent-reported puberty contributed 76% indirectly.
70% of indirect effects included parental puberty with significance. Yeah.
And then for child-reported puberty, 0%.
Zero.
So my question is, from that, can you make any argument as to whether the parent report or the child report of puberty is more accurate? Or is it that this parent-reported puberty may be a report of something else?
Yeah.
Does that lean into that, or is that an entirely separate topic?
It's interesting. So again, going back to the idea of can you say yes or no, the original paper used parent-reported puberty. Based on this, I would say they made the right call. Because, well, what's right call? One of them had 0% indirect effects, one was 76. But you are left with, I made the mistake right there, saying one is clearly better because it had more significant effects. Not necessarily.
Because
right?
the other thing I was going to say is maybe the child report is the most accurate report of puberty, and then it just doesn't have an effect on the outcome measure. So
there's a few things here. I'm actually working on a paper right now with a colleague, Natasha Shaku at Indiana, about the puberty measure in ABCD, because it's the baseline data. They are 9 to 10 years old. As we know from puberty, especially for boys, there's probably not a lot of pubertal development going on at that time. Girls maybe a little bit more, but on the Tanner stage from 1 to 5, you wouldn't expect a lot of variation, especially for boys. And it could be that parents might be more aware of this. Parents might have a greater sense, especially if they've had multiple children, of how long puberty takes for their children, what it looks like. Well, children, although you give them drawings and things like that, I believe in the self-report, they're not necessarily sure about where they are. So in this case of 9 to 10-year-olds, parents might be more accurate responders than children. Is that the case for 14-year-olds or 13-year-olds? I don't know if that would still hold. And one of the things that we can hopefully do in the future with ABCD, one of the things I have to do, is to take a look at that data and kind of compare them a little bit. But yes, you're right. They might be measuring. It's easy to say the child one is error and noise. It might not be. They might be measuring something else. And I do think there's a gender difference there as well. I think parents' understanding of puberty in girls, I think, is probably better than their understanding of puberty in boys. I don't know if everyone agrees with that. So yeah, I'm not sure. But the hybrid didn't also help that much either, but maybe just putting things in half maybe isn't the right answer.
Yeah. And then can I ask my second question? When you talk about frequentist approach and doing multiple analyses, and you talk about p-values, can I ask what your opinion is on p-values and reporting and using the frequentist approach? Yeah,
I think it comes back to, again, that question of how do you communicate this to people? I think p-values are really useful for communicating to people. People like to know is something significant or not significant, and they definitely don't like when you try to give them a Bayes example. They definitely don't like when I give it because I'm not the best at Bayes anyway. But it's a tricky one because we know this. We know that p-values are 0.0501 and 0.04999 maybe shouldn't be treated completely separately from each other. I do agree with people that say, well, you got to have a cutoff somewhere. If you're going to use p-values, you have to draw a line somewhere, and you might as well draw it then and somewhere else. So I'm in two minds of it. I can see the points of significance. I can see how it's useful, but I personally think in this particular multiverse analysis, I mean, I was one of the 5 judges, so I guess I can say this. I didn't use significance a lot. To me, if you can't say something replicated, if it has a p value, which is in both cases below 0.05, but the direction of the effect is not the same. That is not a replicated effect in my book. I don't know if you can say it replicated when the size of the effect, the size of the basis, is very different to each other. For me, significance was the least important of those 3 values that I looked at for me, but not everyone might do it the same way. But yes, it's a tough one.
Very interesting. Thank you. Astrid saying in the chat, fantastic talk, Dom.
Thank you, Astrid. Hope you're feeling well.
Do we have any questions from the audience? Oh, Irene?
Hi, Dom. Nice to see you again. I know Tom, we worked together on a paper. He's brilliant. I have a genuine question. So this is a real question, and I'm trying to I want to pick your brains. I put in a grant recently to look at trajectories of internalizing problems. And in two data sets, the ALSPAC, which is a birth cohort born in the 90s, and the Millennium, which is another birth cohort, both in the UK, both born in 2000s. So I did that because I really wanted to make the most of the data available, but now I worry. It's a genuine worry. People will, I'm sure some of the results will be different. So as a good scientist, of course, you try to make things hard for yourself, right? You really want that's the basis. So I'm wondering, what is the state of the art in psychology, broadly speaking, about replicability across different samples? Well, where actually it's not just different samples in this case, they're different, slightly different periods. So you may think that 10 years is not a long time. Well, I think it is. I think it is for the mental health things that I'm interested in, because a lot of things have happened over the 10 years. So I really think that, people understand and respond to questions differently. So what do we in psychology think about replicating across samples, across periods, across populations?
Yeah, I agree. My instinct, I think it's a hard question I don't really have the answer to, but I think my instinct is that we should have more, in psychology, we should have more papers that are large and comprehensive over multiple data sets. I think there's often a tendency to do one paper about one data set, another about another data set, and not really ever compare the two or combine them. I do think having larger studies with more data sets and you explicitly lay them out as saying, well, I'm interested in this effect across time. I know this isn't an exact sample. This is more of a conceptual replication than it is a perfect replication. But, I've got a sample from the 90s, from the noughties, from the 10s, and I can not only conduct the same analyses and find the same standardized effects in all three, I can see how they've changed over time. And something I think would be great is when we have more replications, is something that looks more like a meta-analysis of replications, where you bring all these replications together and you can put them on a scale of time. Time is your is on your x-axis, and you can see, look, this relationship between, I don't know, the amount of time you spend with friends and internalizing issues is getting bigger or smaller over time, or maybe it's not a consistent pattern. That is how, in my mind, I would deal with this issue of chronological time and the things that has changed over it. But to do that requires a lot of replications, or I guess conceptual replications of similar questions. But in terms of, it's a tough one, but I do think having papers that have multiple data sets, the larger papers, grand works, is the answer, but we all know the publishing machine doesn't necessarily reward those. Sometimes the system is set up to reward lots of small papers rather than magnum opuses. But what do you think, Irene? Well,
actually, in epidemiology, this is an old issue. So people who work across epidemiology know that cross cohort comparison is quite standard, accepted, and it's very valuable. It has its own technical challenges, but in psychology, I think this is kind of new. And the problem I have with my particular grant application is that the questions are not asked in exactly the same way at all. So you have different samples, slightly different time issues, period effects, but also slightly different questions. Right. So anyway, I think it's a very interesting philosophical problem, if I'm honest. And I would be delighted to be given the chance to actually think about these issues if I get the grant. But I really worry about how people will see, because if they find effects that are similar, they will say, oh, yes, that's good. If we find effects that are different, then we don't really have the tools to explain. Yes. But anyway. Yeah, it's an interesting one. And I think that as we as the psychology field matures, these kind of philosophical issues will become much more open and discussed. Yeah. How do we discern meaningful difference from noise? Yeah, exactly. Important question, a difficult one. Yeah.
This is probably
And
where
actually, and can I just say one more thing?
Yeah.
I actually got the idea yesterday. I didn't know about the ABCD paper that you described, but I am about to use ABCD. So I'm excited, actually. Yes, as of yesterday. So happy coincidence. Anyway, that's all for me now. I think I should sort of shut up. Yeah.
Thank you so much, Irene.
Thanks, Irene. Yeah, you raised a really difficult issue we have there. So it's just gone 5. Maybe we can take 1 or 2 more questions if there are any.
Sure.
Yeah, Dom's happy to stick around for another couple of minutes. Sorry, we're getting a bit of echo. Yes, any other audience members have any further questions?
It's all right. I'll pitch again my lunch and learn talk for PhD on the 4th of July at 12 o'clock. Because some of my answers to these questions, to be honest, is we should be trying to do more analyses as specific to each person. Maybe not care about generalizing as much across people, and I talk more about that in that talk. So, yeah.
Do you have a link you could share?
I will have it. I don't have one yet, but I will.
Okay. Okay, thanks everyone for listening. Apologies about the feedback. So I hope you'll join us next week at the same time, where we'll hear from Professor Louise Dye about the effects of diet on cognition and mental health. So thanks again.